Okay, you ready to go over this first page? Or you want a few more minutes? Just the first page. Just front, yeah. Okay. So this first little, it's called a cloze activity, where you're essentially filling in the blanks, but using the context clues from the narrative to figure out what goes where. So we know DNA stands for deoxyribonucleic acid. And how is that different from RNA as far as the name? I mean, ribonucleic acid, deoxyribonucleic acid. What's that major difference? Hence the name change. What? Okay, yeah. So in the sugar in RNA is called ribose. In DNA, it's a ribose, but it's lost in oxygen. Hence the term deoxyribose. And that's one of the major differences. All right, so who identified the structure of DNA in 1953? Very good. James Watson and Francis Crick. They elucidated the three-dimensional double helical structure of DNA. By the way, that makes for a good extra credit question on a test. So DNA is extremely important to the cell, obviously. Genes are composed of DNA and are located on the chromosomes. And we can also say chromatin, and I'll show you what I mean by genes in a minute. And this chromosomes and chromatin are found where? In the nucleus of the cell. Now, in what situation would the DNA be considered chromatin?
It's not in the X shape, it's just like a...
When is the DNA in that X shape, that thickened, condensed, dense X shape? During mitosis, during cell division, mitosis or meiosis, which is the formation of sperm cells or egg cells. So when a cell is undergoing cell division, that DNA condenses into those chromosomes. Otherwise, it's in the form of chromatin, which is this big soupy mess of DNA. When we talk about genes, we're talking about a very specific area within the DNA. A very specific set of bases from point A to point B. And we'll come back and talk about this when we talk about protein synthesis. So a gene is a specific segment of bases on the DNA. And you may already be familiar with this, and unfortunately the video we didn't get to it, but DNA is a set of instructions. Instructions for what? Instructions for making what? Proteins. Okay, so DNA is a set of instructions for making proteins, and the genes are the individual instructions for individual proteins or parts of proteins. All right, so genes determine the traits that an organism will exhibit. The structure of DNA can be described as a double helix. DNA molecules are composed of parts called the basic building block of a DNA and RNA is a nucleotide. And nucleotide consists of what? There's three molecules associated with nucleotide. Okay, a sugar. What else? A phosphate. And a base, a nitrogen-containing base. Some of these bases have 2 rings, some of them have 1. I don't need you to know which ones are which. And what we do then is we will string these nucleotides together. And then, of course, on the opposite side, we have other bases connected to other sugars and phosphates. And what holds those bases together? The hydrogen bonds. So look at DNA kind of like a ladder. And the steps or the rungs of that ladder are the bases. And it's hydrogen bonds that are keeping those bases on one side of the DNA strand and the other held together. Now, there are enzymes that are very easily able to break those hydrogen bonds to allow certain processes to occur, which would include replication of the DNA and copying of the DNA to make proteins, which is what we're going to go over. All right, so DNA is organized like a ladder. The rungs or steps of the ladder held together by weak hydrogen bonds. These bases combine or connect in a certain pattern, which is what we call the law of complementary base pairing. Adenine will always be bonded to thymine. Guanine will always be bonded to cytosine. Bases are always bonded to the blank molecules. So we have our phosphate, and here's our base, a nitrogen-containing group, and it's always bonded to what? The sugar. Now, in DNA, again, that sugar is deoxyribose. In RNA, it is called ribose. And regardless, the base is still bonded to that sugar, and the phosphate is also bonded to that sugar. All right, the base called blank is never found in DNA, but it's only found in RNA, and that would be uracil. The complementary strands of DNA, each side of the DNA molecule, are blank to each other. Anti-parallel, sure, yep. We could say opposites, or we could even say complements. Honestly, I pilfered this from some website. Why reinvent the wheel? Because it did what I wanted to do, so I had to make up the key. I think if I would have made this myself, I would have omitted that one, but anyhow. All right, so the diagram there. Let me put this up on the document camera here. All right, so you're given a key, or hints as they're calling them. And so this is, again, you're applying that law of complementary base pairing. So thymine and guanine. So every time you see a triangular tipped base over here, we would have a thymine, right? Let me know if I miss one. Being this close, it's kind of hard to tell. All right? So if these are bases, what are these here?
Sugar.
Okay, those would be your sugar, and then this would be your, those would be your phosphates. Okay, so kind of a simple line drawing of DNA. All right, now the other one is guanine. It's the concave one, so guanine, 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 guanine. All right, so law of complementary base pairing says that T always pairs with A, and G always pairs with C. So across the top we'd have T, C, A, G, C, A, G, G, A, C, A, right? Along the bottom, right to left, A, G, T, C, G, T, C, C, T, G, T. Okay? And then along the bottom here, you're just given the bases. We want to know what the complement is. Let's see how fast we can do this. See if I can write as fast as you can say them. All right, ready? Go. G, A, C, G, C, C, A, T. All right, let's slow down. Everybody catch up. A, T, T, G, A, C, G, C, C, A, T, G, G, A, T, C, C, G, A, T, C, G, A. Okay. If you understand this complementary base pairing, that's half of understanding protein synthesis. Okay? Because when we look at protein synthesis, we're talking more about RNA now. And RNA does have these bases, and these bases do still match up, except for what? What's the difference? There's no thymine in RNA, we have uracil instead. Okay? All right, so all right, so you've seen a similar diagram before. There's the double helix. This is how the DNA coils around the proteins to form those nucleosomes. And ultimately, as the DNA replication is occurring and mitosis is occurring, that DNA condenses into these X's, and these nucleosomes coil and fold and twist and bend until we get this condensed chromosome structure. All right, so here's our basic structure, and this is a structural diagram of a nucleotide. Here's your phosphate, here's your sugar, and here's your base. Nitrogen-containing groups, quite a bit of nitrogen in there. Here's a cartoon, and these are the different bases. You notice that some of those bases have 2 rings, some have 1. I don't care if you know which ones are purines and which ones are pyrimidines. I don't care if you know which ones have 1 ring or 2. That doesn't matter to me. I'm concerned you understand this idea of the law of complementary base pairing. Okay? Which is we just went through that. All right. Again, showing the phosphate sugar backbone, the bases and the hydrogen bonds between the bases that form the rungs of that ladder. Again, some more practice with the law of complementary base pairing. I don't think we need to go through this again, unless you want me to. Okay. We know that DNA is a set of instructions. And the instructions are for making proteins. We know the DNA is found in the nucleus of the cell. The DNA does not leave the nucleus. Where does protein synthesis take place? In the cytosol, which are found in the ribosomes. Now, if the DNA can't leave, how do we get those instructions from the nucleus into the cytosol where the ribosomes can then make the proteins from them? Well, that's what I'm here to tell you today. And that's where a very specific form of RNA comes in called messenger RNA. It's the messenger RNA that makes a copy of the DNA. It makes a copy of the instructions. Those instructions then, the copy goes in, leaves the nucleus through those openings in the nuclear envelope, the nuclear pores, goes into the cytosol, interacts with the ribosomes, where proteins are then synthesized. Now, I talked about genes. These are specific segments of DNA. Those are specific sets of instructions for a protein or part of a protein. You know, the video I tried to show you talked about the Human Genome Project. Back in the 90s, scientists assumed that there was 100,000 genes in the human genome. Actually, there's only about 25 to 30,000. And so now that they've figured out all the sequence and all the A's, G's, C's, and T's in the order, what they're trying to figure out now is how do all these sequences of bases then produce all of the different proteins in the body? Even though there's only 25,000 genes, there may be 100,000 proteins. So that's the next phase, is then to figure out the human proteome. What are the proteins? How do they form? How do these different genes maybe overlap to form the different proteins? All right, when we talk about protein synthesis, there's 2 major steps. There is the transcription phase, which is basically where we are making a copy of the DNA. This occurs in the nucleus. The translation phase is where we convert the instructions from the nucleotide language to the amino acid language. Translation is the actual process of assembling the amino acids to make the protein. Okay, so let's talk about transcription then, if you don't have any questions at this point. When we look at DNA, we have two strands. We have what's called the template strand. And we also have what we're going to call the coding strand. I thought I fixed that on my PowerPoint, but apparently I didn't. Called the coding strand. The coding strand is the instructions. And the messenger RNA makes a copy of the coding strand. With me so far? So, if this AG, ACG, etc., that's listed there, is the template strand, what would be the complement coding strand of DNA? T, G, C, A, T, C, C, G. So it's this coding strand that is the set of instructions. And the messenger RNA makes a copy of the coding strand. Now, with that in mind, what would the messenger RNA sequence be if we're making a copy of the coding strand of the DNA? We're duplicating that coding strand by using this template. All right, so what would that messenger RNA sequence be? So what's going to happen is the messenger RNA nucleotides are going to come in and they're going to form a complement to the template. So if we have an A, what's the complement base in RNA? U. There's no T. So we would have U. What's the complement to C in RNA? G. What's the complement to G in RNA? C. Now we have two T would be A. Another T would be A. We would have a U, a C, and a G. Now I'm going to go through this again with you. But this is what our messenger RNA sequence would be. Notice that this messenger RNA is pretty close to being identical to the coding strand of the DNA, right? Because we're making a copy of the coding strand. It's like taking a piece of paper and putting it in your copy machine and you get a copy of it. Except what's the difference between this copy and the original? What? Uracil instead of thymine. Okay? Now, enzymes play a pretty big role in this. There's one enzyme in particular called RNA polymerase. And this is the enzyme that breaks the hydrogen bonds, allows the DNA strand to separate, and then allows those individual messenger RNA nucleotides to come in and form that messenger RNA chain. Okay? So here's a little flash animation of transcription. So what we have here is our DNA, our double strand of DNA. And so what we're going to see is that there's our enzyme, the RNA polymerase. That's going to break the hydrogen bonds between the bases. Okay? So breaks hydrogen bonds. All right, so there it is. All right, now we can see that it's breaking the bonds. Our messenger RNA starts to pair up with this bottom strand. So which of these two DNA strands would be our coding and which would be our template? Remember, the messenger RNA is a copy of the coding, and in order to make a copy, the messenger RNA has to pair up with its opposite. The bottom is the template, the top is the coding. Because watch, we want to make a copy of the coding sequence. So let me run this. So here's our messenger RNA sequence right there. So is this messenger RNA sequence a copy of the top strand or the bottom strand? And basically, let's look from here to here. So is it a copy of the top or the bottom? Again, we're putting this in a Xerox machine, as the old folk would say, a photocopier. This messenger RNA is a copy of the top one. So the top one here is our coding sequence. And the bottom one is our template. So in order to get a copy, to make a copy of the coding, the messenger RNA has to pair up with the template. We don't want it to pair up with the coding because then we would not get the right sequence. Yeah? Not necessarily. Without getting into a lot of detail, there is what we call the 5' and 3' and 3' and 5'. And it's based on that determines what's what. I will always tell you, in some way, you would be able to know what's coding. If I give you a messenger RNA and say, what's the coding? Well, you should know that this is a copy of the coding except for uracilthymine. Okay? So in this situation, yes, the top one is the coding. If this was rotated to be horizontal or vertical, then it might be the left or the right. All right, so let's run it again. I'll kind of step you through it. So there's our enzyme. And the enzyme looks for a very specific set of bases. It's called the promoter region. And it knows where to go, and there's probably other enzymes. In fact, I know there's other enzymes and molecules that are attached to the DNA that this enzyme looks for. So the DNA polymerase, or RNA polymerase, breaks the hydrogen bonds. You can see that the individual messenger RNA nucleotides come in, they bind to their complement, and eventually they get joined together to form this chain of nucleotides, which becomes our messenger RNA sequence, or messenger RNA strand. And when the enzyme gets to another certain set of bases, it's called the terminator. I'll refrain from doing my Terminator impression. The process stops. And now the transcription is complete. We have our copy of the template. Notice that as the enzyme moves down the sequence of DNA, the hydrogen bonds will reform, and that DNA sequence goes back together. Okay? So that's the first part. That is the transcription. That is making a copy. So if you were to transcribe what I'm saying here, which you kind of are, you would be making a copy. You're writing down what I'm saying, making a copy. So transcription is making a copy. All right, so here's what I want you to do. Take a couple minutes, and I want you to write a brief summary, write steps. What are the steps in the transcription process? You can break it down into 3 steps, 4 steps, 5 steps, whatever's easiest for you. So write down, and feel free to talk and work amongst each other, with each other, to come up with what you think those steps are in the transcription process. I'll give you the first one. RNA polymerase attaches to the DNA. Okay, I'm not going to write these on the screen here, but I will kind of walk you through the basic steps that you should have. Okay, so first thing you should say is that the RNA polymerase attaches to the DNA. It will break the hydrogen bonds, thereby opening up the 2 sections of DNA. The next thing we can say is that individual messenger RNA nucleotides will come in and pair up with the template strand. This is all being recorded. You can come back and listen and write it down. As the RNA polymerase moves down the DNA, more messenger RNA nucleotides are added. The hydrogen bonds between the DNA nucleotides are reforming. The RNA polymerase reaches what we call a stop sequence or a terminator sequence. And the messenger RNA sequence then is done. Simple as that. Okay, now that's just the first part of protein synthesis. We've just made a copy of that template, or of that coding strand. That's all we've done. Because DNA can't leave the nucleus, we have to find a way to get those instructions to the machinery that's going to build those proteins, that are going to read those instructions. Okay? All right, so the next step is the translation process. And the translation process, as I said earlier, is the conversion of the nucleotide language, that messenger RNA nucleotide sequence. It's converting that from nucleic acid language or nucleotide language into amino acid language, because it's the amino acids that make the proteins. So it's like taking plans on a piece of paper and actually taking those 2-dimensional plans and building them into a 3-dimensional structure. Like if I'm building a cabinet, I'll draw out plans. I'll make a copy of my original plans, bring them in the workshop, and then I will translate those drawings into the actual physical structure. That's what happens here. Okay? So where does this process occur? Where in the cell are we going to see this translation occurring? Or maybe I should ask you, what organelle does the translating? The ribosomes. Some of those ribosomes are free ribosomes floating around in the cytoplasm. Some of those ribosomes are associated with what other organelle? Endoplasmic reticulum. And what do we call that type of endoplasmic reticulum where the ribosomes are attached to that? A rough ER. So where are we going to find the ribosomes in the rough ER? What part of the cell? Cytosol, the watery portion of the cell. So, why does the messenger RNA leave the nucleus? Because the DNA can't. Because the machinery for making and reading the instructions is in the cytosol. But the DNA doesn't leave. So the messenger RNA is able to leave the nucleus. Individual messenger RNA nucleotides can come from the cytosol, go into the nucleus, go back out. Now there is a second type of second type of RNA involved in translation, and this is called transfer RNA or tRNA. Because what it's going to do is transfer the amino acids to the ribosome and allow those amino acids to assemble into a chain. So hence the name transfer. It's transferring the building blocks for making that protein into the ribosome. It's like a delivery service. And this is what it looks like. So here we have our copy of the DNA. This is a copy of the coding strand. These are the instructions for making a protein. I'll just let it play a couple times, and I want you to watch it and see if you can kind of figure out what's going on here. We'll play it again. Any idea what's going on there? Okay, let's take it step by step. So first thing, what we see is that ribosomes have two parts. And ribosomes are actually made of rRNA, called ribosomal RNA. They are actually RNA that's twisted and folded in such a way, made in the nucleolus of the nucleus, and there's two parts to it. And sometimes these two parts will come together, kind of form a messenger RNA sandwich. Or sometimes they're already together and the messenger RNA kind of gets fed into it. So what we'll have then is we could simply say that the messenger RNA enters the ribosome. Okay, here's the large subunit. Now here's what we call the transfer RNA. This is the molecule of RNA that delivers the amino acid into the ribosome to begin to assemble the protein. Now it's the messenger RNA, these bases, that tell the ribosome what amino acid to bring in and what amino acid to assemble. So here we have that that transfer RNA kind of embedded in part of that ribosome. What we're going to see next is that another transfer RNA will come in, and it will form a temporary pairing with this sequence here. What do we call a 3-base sequence in DNA and RNA? It's called a codon, C-O-D-O-N. It's a 3-base sequence. And every 3 bases corresponds to an amino acid. So in order to determine what amino acid gets brought in, the ribosome is basically reading those 3 bases, bringing in the appropriate transfer RNA so that we can then join those amino acids together. So here's the next transfer RNA that comes in. Now what do you notice about the transfer RNA? What does it have on it besides the amino acid up here? It has its own 3-base sequence, and that's called the anticodon. Basically, that is the opposite of the codon on the messenger RNA. That's how the ribosome knows what amino acid to bring in. It knows that this transfer RNA has to match up complementary to this codon here. So that is called our anticodon. Now what happens is that the ribosome will facilitate the formation of a peptide bond between this amino acid and this one. So what type of anabolic reaction forms peptide bonds? A real specific type. We lose a water molecule. Dehydration synthesis. So we're going to get a dehydration synthesis reaction occurring between methionine and lysine. Then we'll see the ribosome shift. We'll see that first transfer RNA leave. And then we'll see a third transfer RNA come in. How does the ribosome know what amino acid to bring in? The transfer RNA pairs up with this codon here. So it matches up the anticodon on the transfer RNA to the codon on the messenger RNA. We'll again have another dehydration synthesis reaction, forming another peptide bond. The empty transfer RNA leaves. It goes back out to pick up another amino acid. Again, a fourth transfer RNA comes in. We get another dehydration synthesis reaction. And then at a certain point, the ribosome knows where to stop reading. It knows where to stop the production of the protein. And it's called a stop codon. And when that happens, we get what's called a release factor coming in. It's another protein of some sort that will then tell the ribosome to stop reading. This amino acid chain will then separate from that last transfer RNA, and everything kind of breaks apart, and we're left with now our protein, our ribosomes, our empty transfer RNAs, and we still have that messenger RNA sequence left over. What do you think happens to that messenger RNA after it's done? It could potentially get read by another ribosome to make another of the same protein, or it could break down into its individual nucleotide monomers again and go back into the nucleus to make another messenger RNA sequence for another protein. So this messenger RNA is recycled. Okay, so what would our first step be? Let's do this one together. Let's write these steps out together for translation. So what would the first step be? Oops, I don't want that. So what could we say for the first step? We could say that the ribosome or ribosomes attached to the messenger RNA, or we could say that the messenger RNA enters the ribosome. Either way, the messenger RNA is going to join up with the ribosome. That would be our first step. then we can say, and I know it's a little different on this animation, but then we could say that the transfer RNA comes in. The first transfer RNA comes in. And it brings in that first amino acid. I think this is an important question to know how to answer. And that is, how does the ribosome know what amino acid to bring in? And I already answered this, but what would the answer to that be? How does the ribosome know what amino acid to bring in? It reads the codon on the messenger RNA, and it finds the appropriate transfer RNA that would match up with that codon, and that's how it knows. If a transfer RNA comes in that isn't a complement to this codon, it won't attach and we won't get the amino acids joined together. So we could write kind of a blanket statement that says, well, we could say that the second transfer RNA comes in, and then we get a dehydration synthesis reaction occurring between the two amino acids. Then we'll see that that first transfer RNA leaves. And a second transfer RNA comes in, or a third in this case. And that process is going to repeat. This peptide sequence will undergo dehydration synthesis with this amino acid to form a longer amino acid chain. Transfer RNA leaves. Another transfer RNA comes in. Dehydration synthesis. So we're stringing those amino acids together. It's like making Christmas time, making popcorn on the string to put on your tree. Anybody still do that, or is that like back in the 1970s? Okay. Or from the 1800s, one of the two. And then eventually this process will repeat until the ribosome reaches the stop codon. And then everything separates. We have our amino acid chain, which can bend and fold and twist and become that three-dimensional structure. Ribosomes split apart. Messenger RNA breaks down into the messenger RNA nucleotides. And we now have our protein, or part of a protein. Now they just did 4 amino acids here, which would be how many base pairs? Or how many bases? 1 amino acid is equal to 3 bases. 4 amino acids times 3 is 12, so there'd be 12 bases. Some genes might be a couple hundred bases. Other genes might be several thousand or tens of thousands of base pairs. Now here's a question I think is important. I guess all my questions I think are important. How many amino acids are there that play a role in making proteins? 20. Why do we use a 3-base sequence? Why do we have a codon that is 3 bases rather than 1 base, 1 amino acid? Say that again? Perhaps. If we had, there's 4 bases, right? if we had one base was equal to one amino acid, how many amino acids could we use to make proteins? If one base was equivalent to one amino acid, we'd only have 4 amino acids we could use. Our proteins would be very limited. All right, so, well, what if we did pairs? AA, AG, AC, AT, and then all different combinations. There's 16 different combinations of 2 letters between the A's, G's, C's, and T's. Why don't we use pairs? There's still 20 amino acids. We would have more diversity in our proteins, but would only be able to use 16 of those amino acids rather than the 20. So, if we do a 3-base sequence, we actually end up with 64 different combinations of 3 letters, which is more than enough for the 20 amino acids. So what does that tell you then about these codons with respect to what amino acids go with those codons? There's more 3-letter codons than there are amino acids. So does that mean that AAA is only 1 and something else, and that we're still only going to have 1 amino acid per codon? What's that? Correct. You can have multiple codons that are going to have the same amino acids. So I don't know, your book on page 104 is a very limited table. Under number 2 there, examples of the genetic code. It says messenger RNA codon. So UUU is equivalent to phenylalanine. Well, there may be another combination. I have it in my notes or not. There may be another combination. Maybe it's UUG. That may also be phenylalanine. So there are some amino acids that have 2 codons. Some amino acids only have 1 codon. Some amino acids have 4 or 6. That we can get the same amino acid for 1 or 2 or 3 or 4 different codons. And it has to be that way because there's 20 amino acids, and if we want to use them all, we have to have enough combinations of the bases to get the amino acids we need. Okay. Second page of the packet, it has a number 1 on it. Looks like this. So let's fill this out. So what we have is we have the DNA template strand, and we have our coding strand, and then we have the messenger RNA, and then we have the amino acid. You can ignore that last one. This is not something I'm concerned we know how to do. Okay? All right, so if our DNA coding strand is TAC, what is its complement template strand? ATG. Good. If our DNA template is ACC, what's the coding strand read? TGG. We'll come back to this one. If our template is GGG, what's our coding? CCC. All right. For these other 2, you're only given the messenger RNA. We know that the messenger RNA is a copy of what DNA strand? The messenger RNA is a copy of the coding strand. We're making a copy of the coding, so the messenger RNA should be identical to the coding except for what? T and U. So, if the coding is TAC, what's the messenger RNA going to be? UAC. Remember, it's a copy. It's the same thing except for we're substituting uracil thymine. If we have TGG, what would be our messenger RNA copy? UGG. If we have CCC, what's our messenger RNA codon? CCC, it's the same thing. Follow me on that? All right, now let's work backwards. We're given the messenger RNA, which is CUC. We know that messenger RNA is a copy of the coding strand. So what would our coding strand be if the messenger RNA is CUC? CTC. And CCU, it would be CCT. Now you fill in the template. Our coding strand is CTT. What's its complement template strand? GAG. And if our coding is CCT, what's our complement template? GGA. Okay? Now, what I used to have students do is I'd give them a table, and then I would have them look up the messenger RNA, the UAC, to figure out what amino acid it is, or the UUG to figure out what amino acid is. CUC corresponds to the amino acid leucine. I'm not going to ask you that, but that's what that would be. Okay? Now you do have a second one that I'm not going to go over with you in class, but what I'd like you to do is work on this at home. Now, here's the deal with this. One of the things that I wanted students to do was to think, if you're only given the amino acid, can you work backwards to figure out what the DNA is? And the answer is yes. So that's what I was attempting to do here. You can skip this column. What I'd like you to work on between now and Monday is to do this. You can omit this amino acid row, but do the other 3 rows and the other 4 columns. Okay? I'd also like you to work on page 133, which is the next page. Page 121. This one you should be able to do. I'm not going to do it with you right now. So what you're given down here, this is your template strand. Maybe you want to write this down. This is your template strand on the bottom of DNA. This is your coding. This is your messenger RNA. Here's your transfer RNAs. So what you'll need to do is determine the coding sequence of the DNA based on the template. You'll then need to determine the messenger RNA sequence based on the, well, you could do it a couple different ways. And then I want you to tell me, what are the anticodons that correspond to those 4 transfer RNAs up there? You don't have to worry about what amino acids go in there. You can ignore that. But I'd like you to fill in the boxes. And then page 127 through, well, it should be 130. So there's 4 pages. It's a transcription translation practice test. I'd like you to work on that too, because that's kind of an application of what we just talked about. Now they do give you some codes for some of the amino acids, which if they're given to you, I'd like you to attempt to figure out how those problems work. I don't think it's super difficult. Okay, if there's a couple things you don't understand, skip them. We'll go over them in class on Monday. So here's what you need to do for Monday. So number 1, complete the packet. see that, can you? Oops. So complete the packet. The very last page is a concept map that kind of outlines the whole process. And I will maybe take a little time and go over that with you on Monday. So complete the packet. It shouldn't take you very long. I'd also like you to read pages 110 through 109. No, that doesn't make sense. 104 to 109 in your textbook. You do have notes in your course pack that go with those. And those are the pages that your book talks about as far as transcription and translation. So I think it would be important to reconcile what I've talked about here with what the book has to say and see if you can put that all together. It's the same stuff. I wanted to kind of give you a basic primer first. So I want you to read those pages and finish the course pack for those pages. And then you'll have content area 2.3 to work on. And I'm going to limit how much stuff is in the next content area, because I didn't have a chance to go over any tissue stuff today, which I think was on this content area, and some transport processes. So what I think I'm going to do is I'll have a very short content area for Monday to cover for you to prepare. Make sure you do this. I'll answer any questions you have about protein synthesis. We'll go over the packet. I will go over the notes and things that you had to prepare for today. And then I'm going to do a little lecture on cell-to-cell connections. I'm going to be gone on the 28th. I have a conference in Washington, D.C. I have to go to, that I'm going to. So we will not I think what's going to happen, I'm going to move your exam up. And instead of having 3 chapters on your test, it's only going to cover chapters 3 and 4. Okay? So I was going to originally have your exam scheduled for the 1st, which is Wednesday, but I'm going to be gone those 2 days. I won't have time to review anything with you. So I think what we'll do is we'll have our exam on the 28th, is that right? 29th, sorry. Monday the 29th, and I'll put this in an email. I'll be gone that day. So on Monday the 29th, you'll have your exam over chapters 3 and 4. And we'll wrap chapters 3 and 4 up by next Wednesday, okay? And then I'll just add chapter 5 on to the next unit. We'll have to kind of, I don't want too many chapters on one test, so I'll have to find a way to reconfigure things. Maybe I'll have you do part of the test on Blackboard or something for chapter 5, okay? So I will get that content area ready to go tonight or tomorrow morning, so you'll be ready to do things and have everything ready to go for Monday. Have a good weekend.